The first historical discovery you find about the lymphatic system was documented in 1622 by Italian physician Gaspar Selli. And he, upon dissecting freshly fed dogs, found what he called the milky veins, which was basically the fats that were in the diet that were picked up by the lymphatic lacteals in the small intestine and carries fats to the liver where they can be processed. We note them especially as they're associated with mucosa. So in the intestinal tract, we have Peyer's patches, and the appendix is a lymph node. It has a lymphatic structure to it, we should say, probably not specifically a node. We have nasal associated lymph node tissue, think adenoids. Innate and adaptive immunity are subtly different, and each has cellular components and each has soluble components. Innate immunity is basically the infrastructure of our organisms. Your skin is part of your innate immunity, keeping stuff out. The phagocytes and dendritic cells that rove around in the periphery of our body, part of innate immunity. They're already there. Something didn't have to be mobilized. Something didn't have to be brought to that area. They were there. They're doing their thing. IgM antibodies are the biggest of all of the antibodies. They consist of clumps of other antibodies, and they are the very first antibodies to appear in response to initial exposure to a microbe. Baptisia tinctoria, wild indigo, was a very specifically indicated plant for the eclectics at the turn of the century. It was established by them, we learn about this from Felter and King, that it was a lymph mover with alterative qualities.